Hello, welcome to my presentation, Developing Analytical Methodology and Test Apparatus to Study Drug Deposition in the Throat. My name's Matthew Potts. I'm a Senior Analytical Scientist at Kindeva Drug Delivery, and this is my presentation on the paper I wrote for the Drug Delivery to the Lungs conference. So why study drug deposition in the throat? Well, mouth and throat deposition is the biggest reason for MDI and DPI device inefficiencies. In some cases, over 70% of the dose is deposited in this region, so anything we can learn about the origin and the location of this drug deposition may enable us to develop products with increased efficiency, resulting in low manufacturing costs and potentially fewer or less frequent dosing for patients. Another reason is that the conventional compendial lab testing techniques such as ACI or NGI coupled with a USP throat rarely match real-world or clinical product performance. So any apparatus that could be developed to provide a more accurate assessment of mouth and throat deposition and the product fine particle dose characteristics would be beneficial when developing a new product. The patient experience is also an important factor to consider when developing a product. Any discomfort experienced when using the device is obviously undesirable and may discourage a patient from conformance to the dosing regime. Unwanted side effects such as oral thrush may be minimized or avoided through greater understanding of the regional deposition in the mouth and the throat. The location of drug deposition could also be critical to informed device design and formulation development activities. But the main reason that we want to study the drug deposition in the throat is to result in better products and to deliver better patient experience. So what are people already doing to study this deposition? Well, there are already a number of techniques for studying oropharyngeal deposition. We could use gamma scintigraphy. However, this typically requires human volunteers and a clinical setting with expensive equipment, and therefore is not really suitable for rapid screening or iterative product development testing. We also have to factor in patient training regarding device handling, inspiration rates, actuated timings, which could also be highly variable. We would need to consider any variability in the airway geometry of the volunteers and how if it is representative of the target demographic for the product. Another alternative could be to use computational fluid dynamics to model expected drug deposition. This, however, currently requires a great deal of setup to develop the model, considering all the various potential variables and contributing factors. It can require a great deal of computing power and time to perform the calculations, and still requires confirmatory in vitro and in vivo testing to confirm the model. So again, not that well suited to rapid product development activities or screening activities, although it could become more viable in the future as this technique becomes more established. The next and most common way to assess drug deposition in the throat is to use one of the many commercially available anatomical throats, in conjunction with a compendial impaction technique such as NGI. Models such as the Oropharyngeal Consortium and Virginia Commonwealth University throats provide an excellent way to assess overall throat deposition, but are not designed for regional drug recovery. The Alberta idealized throat allows an easier access to the internal surfaces for drug recovery. However, that idealized geometry could result in some inaccuracies when assessing drug deposition locations. So what equipment do we need? Well, the goal is to quantify regional drug deposition in the mouth and the throat. Therefore, a sectionable anatomical throat would achieve this. This would enable us to develop an understanding of the origins of any oropharyngeal deposition and gain some location granularity. Coupling this with an existing aerodynamic particle size distribution test apparatus would allow us to assess important product performance characteristics such as the resulting fine particle fraction of the delivered dose at the same time. Unfortunately, there is nothing currently offering that functionality on the market, so we decided to develop the apparatus in-house. So we started to develop the KDD split OPC throat, basing the design on the medium-sized OPC throat geometry. We then split the geometry into 4 regions of interest, so that being the tongue, shown here in pink, the oral cavity, shown in green, which ends at the uvula, the oropharynx, the yellow region, continuing down to the epiglottis, and finally the laryngopharynx, which is the blue region consisting of the remaining throat geometry. The design of the apparatus was developed in-house using CAD software, and early prototypes were created using additive manufacturing. This helped us to trial designs and assess them for usability, by which I mean the complexity of assembly and disassembly of the apparatus during testing, and assess the ability to perform successful leak testing on the system. We chose to manufacture the final device using the same materials and manufacturing processes as the commercially available OPC throat from Amaze Consulting. This ensured that there were unlikely to be any specificity or drug recovery impact on any existing product assay methodologies that we chose to use. And all of this resulted in what you see here, which is the design for the KDD split OPC throat. So we have on the right hand side the 4 main components as discussed on the previous slide. We also have a gasket which sits between the laryngopharynx and the assembled top 3 sections, and a second seal situated in a groove in the oropharynx component, which enables us to achieve a gas tight seal for the apparatus once assembled. It's worth knowing that a nitrile sleeve, not shown here, was also later added to the device in order to aid with consistent and successful leak testing prior to use in an analysis. We then needed to couple this new apparatus to an appropriate APSD testing technique, suitable for the intended prototype device and formulation screening activities. We wanted a fast, focused methodology. Conventional APSD testing using NGI or ACI apparatus often requires significant analytical resources, producing large numbers of samples, typically analysed using an LC system. Therefore, it is important to ensure that the methodology is focused on determining the critical data for understanding the problem. In this instance, we were only interested in the sectioned throat deposition and the fine particle dose. Therefore, abbreviated impactor measurement techniques were considered. We chose to move forward using a fast screening impactor with an appropriate insert, minimizing any additional sample preparations whilst still producing the same fine particle dose result. Another option to reduce sample analysis timelines and solvent usage was to use a UV spectroscopic analysis instead of the more common liquid chromatography analysis. However, this option is limited by specificity considerations and may not be viable in all cases. Here is shown the experimental setup with the FSI and the sectionable Amaze throat here in the centre, coupled with a standard high capacity pump and an electrostatic fan. For the case study detailed in our paper, we chose to test a tiotropium PMDI product using both the KDD split OPC throat coupled with an FSI, the results shown here in blue, and compare that to results generated using a medium OPC throat coupled with a standard NGI, shown here in green. Although we observed a small reduction in total drug deposition in the split throat when compared to the OPC throat, the total drug deposition is still broadly comparable. There is also increased variability observed between test replicates on the split throat. However, this is perhaps unsurprising given the additional complexity of the recovery process and for the increased potential for cumulative error. There is also an opportunity to further optimize this methodology in relation to the drug recovery process and use of coating solutions. The mass balance and fine particle fraction results were also very comparable to the conventional testing results, indicating that the split throat is acceptable to use as an indicative fast screening tool. In this instance, we learned that approximately 85% of the total throat deposition was on the tongue, which informed future product development activities. The high drug deposition observed in the throat agrees with previous published work concerning oropharyngeal deposition of aerosolized particles. However, this technique is undoubtedly faster and less costly when compared to gamma scintigraphy for the purposes of quantifying regional throat deposition. I'd finally like to acknowledge my colleagues with which I collaborated on this project, who were all invaluable in their contribution in developing and demonstrating the split throat apparatus. That concludes my presentation. If you have any questions, please do not hesitate to contact me and I'll do my best to answer them. Thank you for listening.